And we're really excited and thankful to have Mary Marovich, Dr. Mary Marovich, speak to us. She is the Director of Vaccine Research Programs at the Division of AIDS and NIAID at the National Institutes of Health. And Mary will be updating us on the national COVID efforts, variants, immune protection, vaccines, etc. So Mary, please take over. And I guess Wendy's going to share the screen. Is that right?
Yeah, I'm going to share it now. So please let me know if everyone can see it.
Looks fine.
Okay.
Okay, thank you very much. I'd like to thank Dr. Sokol and for the invitation to come back and update everyone on the national COVID efforts. Last year when we had this meeting, we were hoping that the meeting this year could be face-to-face, but not the case. Looks like we have a great meeting today virtually, a great lineup of speakers. So if you can move to the next slide, Wendy, I'll give my overview. So I'm going to try and cover a lot of territory today. You'll remember the federal COVID-19 response was originally called Operation Warp Speed. It is now morphed into the federal COVID response. I'll talk a little bit about the clinical network that conducted all the phase 3 efficacy trials, the coronavirus prevention network, and of course mentioning that investigators at the University of Colorado are members of this network and of course made critical contributions to our efforts in the COVID response. I'll talk about the vaccine candidates. I'll talk about factors that impact vaccine efficacy. I'll touch on immunocompromised populations and then move to monoclonal antibody prevention and early treatment and close with variants. So next slide, please. Right, so not too long ago in January, you'll remember Chinese researchers had published the genome of the virus that was implicated in the Wuhan pneumonia outbreak. And within about a week of that, next slide, the Vaccine Research Center with Barney Graham and Kizzy Corbett, next slide, please, were able to use their experience on prototypic pathogen from the coronavirus family. They had experience working on SARS and MERS, et cetera, to use that for vaccine development for SARS-2. Next slide. And the point here that I want to make was that they used structure-based vaccine design, which has been used for other vaccines, RSV, et cetera. Barney Graham has been one of the leaders in this field. And basically, as you'll see on the left is a cartoon of the coronavirus showing the envelope spikes. And then in the middle is an image of SARS-CoV-2 virion. And it shows sort of this irregular corona halo. And those are actually spike proteins. They're in the pre-fusion form. And as you can see, they're somewhat irregular because it's an unstable form. And so Dr. Graham and others realized that they needed to stabilize that pre-fusion form to make a good immunogen. And they introduced a couple of mutations, proline, that's the S2P designation, stabilized the pre-fusion form, and then developed the mRNA vaccine. Next slide. Right, so within, as you can imagine, within a couple of months, they were able to produce the vaccine in collaboration, of course, with Moderna. And they started a phase 1 study that started small, enrolling 45 participants, and then gradually moved into the efficacy trials that I can talk about in a second. If you go to the next slide. Right, so as we were dealing with the epidemic, we realized that we had to harness all the talents and attributes of the US government, academia, and industry to collaborate in unprecedented ways. And We designed sort of a roadmap on how we would develop the countermeasures, vaccines, et cetera, to fight the epidemic. And this was published in Science in May, and it was our approach to vaccine research and development. Next slide. And I'd like to show here is just the USG component of the federal response, which was referred to Operation Warp Speed, as you'll recall. And it was really made up of the HHS and the Department of Defense. And from HHS is BARDA, the Biomedical Advanced Research and Development Authority. There was NIAID, there was CDC. And then from the Department of Defense, there was a joint program, Executive Office for Chemical, Biological, Radiologic, and Nuclear Defense, so JPEO. And then, of course, the Army Logistics Command and other contributions from the Department of Defense. Next slide. And so what happened? We were given an enormous amount of funding to tackle the pandemic, over $12 billion. And the types of efforts that funding was used was obviously to work on vaccine research and development, which included the conduct of these phase 3 vaccine trials. All of them are ongoing, and participants remain in follow-up. Also, much of the funding went to commercial scale manufacturing, and that was, of course, handled by BARDA. That's an area of their expertise. And then, of course, there was advanced purchase agreements made. You'll remember purchasing 100 million doses of Pfizer, for example, before even it was authorized. In the event that there was going to be successful vaccines, the United States wanted to make sure that we had those available. And that was what we sort of got in return for our investment. So next slide. Right, so these are the 6 sort of priority vaccines that we focused on. They're in the portfolio. And you'll remember there's 3 different platforms: the nucleic acid or the mRNA platform, then the viral vector, and then the protein subunit itself. And we took 2 from each of those different types of platforms. Moderna and Pfizer were the mRNA vaccines, as you well know, and there's a little picture of the mRNA here. And basically mRNA technology had been around for quite some time, but the issue was trying to stabilize it and encapsulate it in a lipid nanoparticle that was suitable for delivery. And they were able to do that. And as I mentioned, incorporate the stabilized, the VRC stabilized prefusion immunogen. And I'll show you the results in a second. Then the viral vector did the same. It inserted that message into the viral vectors. And then, yeah, just really quickly here. So then the mRNA is really fast because you synthesize the mRNA and then encapsulate it. So it's very efficient and it's very adaptable. The viral vectors are sort of intermediate in that sense. They use a harmless, non-replicating virus, like a sort of quote unquote cold virus, adenovirus, as an example, and they insert that same message, that same recipe is inserted into that viral vector backbone, and again instructs the body to make the proteins, and uses one of those, the Janssen used the VRC design. AstraZeneca used a wild type envelope design. And then finally, the 2 companies that used the sort of tried and true standard way of actually synthesizing that trimeric spike protein in manufacturing facilities, which is really complicated and takes a long time. And then combining it with adjuvants, those were Novavax and Sanofi, and both of those trials are ongoing as well. Sanofi is still in the early stages of its phase 3 study. This is organized also in the sort of timing, as I mentioned, because of the speed with which you can develop the mRNA vaccines. Those have become a hot topic. You'll hear a lot about that. And then increasing complexity as you move down the list. Next slide. Okay, and then as I mentioned, we had developed the COVID-19 Prevention Network, which was a virtual collaboration of all the different NIAID-funded clinical research units, the Vaccine Network, the Infectious Disease Clinical Research Consortium, the HPTN Prevention Network, and Department of Defense. And those were divided into two groups. They were led by two co-chairs, Kathy Neusel and Larry Corey, led the vaccine efficacy trials, and Mike Cohen and David Stevens led the monoclonal antibody prevention trials. Next slide. And this is a graphic just to give you a quick overview of how the studies were designed. They were all independent, separate studies, but they were harmonized. And that was part of the tenets of the USG strategy. And they were of course randomized. So you'll see on the left that there was a vaccine group and a placebo group. Normally it was 1-to-1. A couple of the studies did 2-to-1 vaccine to placebo. About 30,000 to 60,000 participants were estimated to be needed for each trial. Of course, the volunteers and the investigators who were working directly with the volunteers were blinded. They were closely monitored after vaccination as they went throughout their lives. They developed symptoms of COVID. They came into the clinic. They were tested by PCR. And if that was verified, they were determined to be a case, a quote unquote case of COVID. And they were tracked accordingly, of course, clinically towards resolution of that disease, but they were noted and counted those cases in the vaccine group versus the placebo group, as you can see on the right. And that was what was tracked as far as how we understood how efficacious the vaccine was, if there was a reduction in the number of cases in the vaccine versus placebo. Next slide. And so those efficacy results started coming in November. And you'll remember that this data was presented and made publicly available through the VRBPAC at the FDA. The data that was submitted to the FDA for consideration for authorization was available and publicly attended. It was found remarkably that there was a vaccine efficacy of 95% for the Pfizer vaccine after a two-dose regimen. And shortly thereafter, in fact, a week after, the Moderna vaccine demonstrated similar 94% vaccine efficacy. And I just have to take a moment here to mention that those are highly efficacious vaccines. That was a remarkable result that many of us, including myself, were pleasantly surprised about. Those studies, the Moderna vaccine was conducted purely domestically. The Pfizer vaccine was done in a few countries outside of the US. But the Janssen vaccine, which is shown last year, which has a few notable differences. One, it was a single dose. It is much easier to deliver. It's much more stable. The RNAs, of course, are less stable and they have to be kept at very cold temperatures. But the adenovirus vaccine is quite stable, can be stored in the refrigerator, easily transported. But it was also tested very widely geographically and was also the first pressure test for the variants that had developed, that began to emerge in December when the other studies were completed. So the Janssen vaccine had to contend with the beta variant in South Africa and other P variants in Brazil, for example. And the overall efficacy, which was combined from all these different areas against all these different variants, was 66% vaccine efficacy for prevention of symptomatic COVID. And of course, the efficacy was much higher for hospitalizations and death. But the US vaccine efficacy was 72%, somewhat higher. We were not dealing with those variants at that time. And the EUA was issued in February. Next slide. So just a quick graphic on the types of data that we follow. I'll just show you on the upper right-hand side. The vaccine efficacy for the Moderna vaccine was published in the New England Journal. And this is the type of curve that we monitor called the Kaplan-Meier curve, where on the x-axis, it's the days since randomization after receiving the vaccine. And on the y-axis, we talk about the cumulative incidence of COVID over time. And what you can see is that the number of cases in the mRNA arm were much lower than those cases in the placebo group, which allowed us to determine and estimate the vaccine efficacy of about 94%. Next slide. So what are some of the key factors that influence vaccine efficacy? This is a great paper. I'll refer you to it in Nature Review Immunology by Tregoning et al. It talks about the progress of all the vaccine development and touches on a lot of these different areas. But For today's purposes, I'll just go into a few of these areas. Of course, we know that the correlates of protection, which are defined by what elements of the immune response would confer protection from the vaccine. Some of the immune factors, of course, that are elicited by the vaccine would be the antibody titers and the quality of the immune response on the lower right-hand corner. Those would definitely influence the vaccine efficacy, obviously. In green is those that have a positive effect on efficacy. Vaccine access factors, which I know there'll be some discussions later today on access, is, depends on which part of the and if you have access to vaccines, which ones are used, the number of doses, and the interval between dosing can be very important. Longer intervals can tend to provide better immune responses and increased levels of protection. There's demographic factors, of course. If there's high levels of circulating virus in the community, if people are living closely together and not social distancing, et cetera, that can, of course, increase, can impact vaccine efficacy. And then there's host factors like age. Increasing age has, of course, been associated with less efficacy or less durability of efficacy of the vaccine. Prior infection, of course, has been associated with conferring protection against reinfection, at least for some time. And then there's the element of the immune system. If people are immunocompromised with the underlying health conditions, as I mentioned, I'll talk about that later, that can lead to less efficacious vaccines and can get those folks into trouble. And then, of course, there's the variants, which can create a mismatch, obviously, between the vaccine and the circulating virus vectors and can cause problems and reduce efficacy. Next slide. Okay, so I'm just keeping careful, close on time here. If you could give me a heads up when I have like 5 minutes left, I'd appreciate that. So there's a couple of papers I want to draw your attention to as I get through the remaining slides here. This is a preprint paper from Peter Gilbert, who is one of the premier scientists in statisticians in analysis of immune responses. It's an immune correlates paper where they basically, as part of the Operation Warp Speed federal response, there was a group of federally funded and NIH biostatisticians who will analyze each of the vaccine results and look at correlates of protection across all of these studies. And what they do generally in this study was they looked at antibody responses, because those are typically associated with protection. And they study the people that are studied are those who received the vaccine. So you take the people who received the vaccine and were infected or were developed COVID. And remember, there were very few of those for these vaccines that are highly efficacious, maybe 38 or 40 cases for this correlates protection. It's not a lot of cases. And then you match those with people who received the vaccine who didn't become infected, of course, because you want to know what the difference is between those who received the vaccine and became symptomatic versus those who didn't. And they focused on binding antibody levels and then functional antibodies called neutralizing antibodies. And the long and the short of it is, you can read the paper, is that the vaccine efficacy is likely linked to the level of neutralizing antibodies. And it's proposed as a surrogate or a correlate. It is supported by, in the table below, you'll see all these different lines of evidence that would support neutralizing antibodies as a correlate for protection. It is not fully established. There are other parts of the immune system that are surely contributing. There's no threshold that's established for the neutralizing antibodies that is thought to come. But it is now being discussed as a strong candidate for a correlate. Next slide. So what does that mean? That means we can begin to look at the immune responses and correlate that with vaccine efficacy of different trials and in different hosts, as we talked about. So how do people with cancer or organ transplants, how do they respond to mRNA vaccines, for example? Well, this is a response rate, so a percentage of responders. You can see most of the folks tested here in a small sample size, 50, but most of the folks on hemodialysis did respond. Most of them were responders. Some some did not respond as well in cancer. Organ transplant recipients, clearly there were a lower percentage of responders to the 2 mRNA vaccines. And if you go to the next slide, this is what has been the driver behind this concern with the immunocompromised and SARS-2 infection. I wanted to go into this because it's a big topic now in the news and in our country and globally. So at least for the United States, there's about 7 million people who are considered immunocompromised. It's only about 3% of the population. However, this group sort of has an outsized role in SARS because they are much more likely to get severely ill if they develop COVID, and that they're higher risk for prolonged SARS-CoV-2 infection because their immune system isn't able to handle the virus as well, which leads to prolonged shedding of virus and allows viral evolution during the infection. They have lower antibody responses. They're more likely to transmit infection to household contacts, and they're more likely to have breakthrough infections. So it's a couple of studies here that have shown that about 40% of hospitalized breakthrough cases are immunocompromised individuals in the US and Israel, for example. So there was a big effort, of course, to protect these vulnerable people. And the idea was that perhaps they'd benefit from a 3rd dose. And what I'd like to show you here is one study that was published. This was a randomized study of about 100 transplant participants who were randomized to just receive the placebo group, just got the 2 doses of Moderna, the regular regimen, the primary. And then the mRNA group here got the 3rd dose. And you can see in all areas of the figure here is that those who had received the 3rd dose of the mRNA clearly had a better response. There were more levels of antibodies. There were more people who developed neutralizing antibodies. There were better T-cell responses. And in aggregate, the FDA reviewed data well beyond this trial, but this is an example. If you can go to the next slide, they did authorize, as Dr. Sokol mentioned, August 12th, the FDA authorized an additional dose for certain immunocompromised individuals. And then subsequently, as what usually happens, then the ACIP, the Advisory Committee on Immunization Practices associated with the CDC, held a public meeting, as they do, hold these emergency meetings and discuss this, and made a recommendation for the individuals that are shown here in bulleted shorthand who could receive, would be eligible and should receive an additional dose of vaccine. And they're outlined here: organ transplant, stem cell transplant, people with primary immune deficiency, advanced or untreated HIV infection, or those treated with high dose corticosteroids as part of their immune response. So it is interesting to note that prednisone is a big player in the immune suppression for those folks who are on transplant, who are transplant patients who are on other potent immunosuppressants. The idea is to talk this through with their healthcare provider, but many people are opting to get a 3rd dose. Next slide. All right, and so what happens to those folks who, despite being vaccinated, they're immunocompromised and they end up still getting COVID, or they're not vaccinated and they get COVID? And there's a lot of risk factors like increased age or diabetes or other comorbidities or BMI greater than 30, various things that put people at an increased risk for complications. They may end up in the hospital, they may end up in the ICU. So as we were developing vaccines, we also thought, well, we could isolate, very potent neutralizing antibodies that could work as therapies in the meantime as we developed these vaccines, not knowing that we were going to have these terrific vaccines that would come so quickly. But there was a great effort to isolate and understand which vaccines protected or could prevent infection. And these are just 2 studies that were done that showed both early treatment effects and prevention effects of COVID in people who were living in long-term care facilities. On the right, the Lilly antibody was tested in residents and staff of skilled nursing facilities where there was an index case in the facility, and then people were randomized to receive either placebo or antibody. And then there was the household contact study on the left, the REGEN-COV antibody, which is a cocktail. Again, people who were living with an index case, if they were presented within 4 days of the diagnosis of the index case, they were randomized to either receive placebo or antibody. And the bottom line here is that there was a dramatically positive effect. There was a relative risk reduction of about 80%. So people, there was a great reduction in developing COVID. There was a reduction in hospitalization. There was no deaths in those treated groups. There was that was related to COVID. There was actually a reduction in development of asymptomatic infection, so acquisition of infection. And the idea was that these antibodies could act quickly, and they could prevent in the setting of exposure, prevent infection or disease, and provide early treatment. But the caveat with monoclonal antibodies is that they work terrifically, but they are susceptible to mutants because the mutants can escape those antibodies. Whereas a vaccine, you have a polyclonal immune response and you have cellular responses that you harness the entire immune system. But with these antibodies that are good for treatment and targeted prevention, they can be rendered less effective by escape by the virus. And that has happened for the Lilly antibody. So what are we doing to look at these variants? Next slide. Just to let you know, I'm closing up now. The United States and others, of course, globally are tracking the variants. There's a SARS-2 interagency group that's comprised of all different elements of the US government, the CDC, the FDA, the NIH, BARDA, DOD, that are basically tracking and characterizing these variants and then determining their impact on countermeasures, which include vaccines, therapies, and diagnostics. This is a good website if you're interested in looking at this. What I wanted to show you is on the right-hand side is something called Nowcast, and it's a model that the CDC developed that provides a weekly snapshot of the proportions of the variants that are circulated in the United States. And as you can see on the x-axis from May to July, the end of July, these are the weekly reports. And in the light orange is the B.1.1.7, which is the alpha variant, which was predominant. And then over a very short period of time, you can see the explosion in Delta, where it just predominates now as most of the circulating virus in this country. And the key areas of investigation are transmissibility of these variants, pathogenicity, do they cause more severe disease, do they evade immunity, and all different types of surveillance. Next slide. So just wanted to highlight that there are these variants of concern, which are, of course, obviously the most worrisome. This is a table that provides a good description of their naming and sort of their attributes. I want to just point out two of the most worrisome variants. That's the beta variant that was isolated in South Africa because it had increased transmission, but it also was the least sensitive to the neutralizing antibodies that were elicited by vaccines or by infection. So it took more of those antibodies to protect. And the next one is the Delta, of course, that's the most current. That has the very high level of transmission. It is very infectious, and it was easily could take over. It was in our country that preceded that, but it's not as impacted by the antibodies, so it is still considered neutralizable. I'll show you in the next slide, but it does require somewhat more or slightly higher levels of the antibodies for neutralization. Next slide. So here's this is a kind of a tricky graph, but the higher the dots are on the graph is the worse it is. So if you look along the x-axis at the bottom, we have all the variants across the bottom, and there would be no impact if it was on the axis here by different variants. But as you go up this y-axis, that shows the fold reduction in neutralization activity, means you need more antibodies, more potent antibodies to neutralize that variant. And as you can see, the beta variant is the trickiest and the most challenging to neutralize. You need higher levels of antibody. If you look next to it is Delta. Again, the remaining variants are about, say, 2 to 3-fold reduction in neutralization activity, but that's something that these antibodies can have the capacity to absorb. If we get those antibodies up high enough, we can protect against those. Next slide. So I'm wrapping up now. I hope we have time for a few questions, but this is a paper I wanted to let you know that was published a couple of years before we had this epidemic. where the US government had, of course, been working on novel technologies and trying to preemptively establish information on various pathogen families and to develop a prototype so that we could have strategies to fight those. And the point was that we need to continue to invest in infrastructure and global surveillance to expedite the identification of these potential pathogens and jumpstart the process of vaccine development. And with that, I think that is my last slide. I'd just like to acknowledge, really, I'd like to acknowledge all the clinical trial participants in all the various trials, the clinical researchers and the support staff who made all those studies happen. NIAID senior leadership, Dr. Fauci, obviously leading us through this pandemic and also kindly allowing use of some of these slides. Carl Diefenbach, Emily Alberting, John Mascola, Julie Ledgerwood, Heather Scobie, and Tina Tong for assistance with presentation. The COVID networks, as I mentioned, the industry and academic government collaborators, sponsors, and developers, Dr. Sokol for again inviting me to give you an update, and the audience for your attention today. And there's a few key links here for you. So thank you very much for your time.
Thank you so much, Dr. Marovich. We do have time for a question or two. So there was a question, and I don't know if you covered this precisely. The question is, have there been any comparative studies on breakthrough infection rates among the various vaccine types? You showed us antibody levels. Do we have data about breakthrough infections?
Yeah, that's a great question, and a very hot topic of interest. Those studies would probably fall into the category of real world type studies. And I know there's going to be a session on that later. But this would be where you'd need large numbers of people who had received, say, for example, Moderna versus Pfizer. There are many studies that are published out there. And that's in fact how we're tracking vaccine efficacy to see if it's drifting downwards. There was some early evidence from Israel that vaccine efficacy was waning to the Pfizer vaccine, which was the one that was mainly used in Israel. And then as we began to look at areas and comparing areas that used both Moderna and Pfizer, we were able to see not as much waning with the Moderna vaccine, although that was slightly behind the Pfizer vaccine. So it's a critical tool to look at comparative efficacy across real world studies as these vaccines are rolled out.
Thank you. Another question. We hear about a possible recommendation for a booster vaccine for everybody.
Yeah.
I don't know if the CDC has officially made that recommendation or the White House. I wonder if you could update us on where that stands.
Yeah, so this is, of course, something that we've been looking at for a very long time. And of course, we wanted to make that decision based on science. So we had to design studies that looked at and compared boosting in people who had received the primary regimen. And then we looked at different vaccines and different combinations of vaccines, and all these studies are ongoing. We are beginning to get the results of some of those studies, those boosting studies that were conducted within the NIH, and should have more complete results towards the end of this month. But it is going to be, I believe, announced by President Biden this afternoon. I believe he'll have a press conference about boosting doses. It does look like there's going to be a recommendation for a third boost to be made available to those who have received the mRNA vaccines. It makes sense that those who received the vaccine first would be further away from their last dose, and those would be elderly who tend to have less good of a response to the vaccine, potentially less durable, and healthcare workers who are exposed to high levels, they're in higher forces of infection environments, et cetera. So we'll get more information on that third boost, but it is likely to be beyond the immunocompromised that was released last week, and we'll learn more information. I'll learn that as you do.
Great, thank you. And one final question. Since antibodies are likely associated with protection, do the people who have symptomatic vaccine breakthrough infections present with low levels of antibody or neutralization antibodies? In other words, are the antibodies levels related directly to breakthroughs?
Precisely. That is exactly what it is. So of course, you might be thinking, well, is there an element of quality, right? Are there certain antibodies that are better? And yes, there are. And that antibody immune response can mature over time. So the antibodies get better and increase avidity, et cetera. But to be straightforward about it, yes, the levels are correlated with protection. That's the basis of the immune correlate study that I referred to by Gilbert. And there's also other studies. There was a New England Journal paper that was just shown in healthcare workers where they looked at what's their antibody just before they developed infection, their level, and then those who didn't, and they showed a decrease in that as well. So as the correlate data emerges, you'll see levels playing a big role.
Okay. Well, thank you, Dr. Marovich. We really appreciate you taking the time to speak to us. And if you'd like to stay on the webinar, we have a bunch of great talks following yours.